This is a production of Cornell University.
Today, I will be talking about my PhD, part of my PhD work, and like we have how we have isolated this P periaculum mutant and how how the folate is regulated in this mutant. So first of all, I want to give you a brief introduction about me. I was born and brought up in near Delhi, like 100 kilometers from Delhi, and I did my graduation from. So then I did my graduation from Merit College, Merit in Zoology, Botany, and Chemistry. And then I moved to Tezpur University, which is like northeast part of India. And I did my like MSc in biotechnology with major in plant biotechnology. And then I got an opportunity to work in the University of Hyderabad as a doctorate. So my PhD, I did mostly on to identify mutant defective in blue light signaling. And we use like forward and reverse genetics technique to characterize them. And then I got a position in Israel in Volcani Center, and I did like collaborative work on in grape. So with this brief introduction, I just want to go back to folate. So why folate are important? So folate, the most of the staple food, they are very poor in folate, as you can see here, like corn, rice, wheat, potato, and cassava. They are very, very low in folate. So therefore, folate deficiency is the worldwide health problem, which affect millions of people. So the recommended diet allowance is 400 for the adult and 600 for the pregnant woman. And the folate deficiency can cause neurological defects, which include spina bifida, like the spinal cord and spines and spinal cord did not close properly, and the anencephaly, where part of the brain is not developed properly, and many other like megaloblastic anemia and high risk of cardiovascular disease and certain type of cancer. So there are some of the rich sources of folate, like green leafy vegetables, asparagus, broccoli, and fermented food products. They are very rich source of folate. So coming to the structure of folate. So tetrahydrofolate and their C1 derivative, also known as folate. They are also like water-soluble form of vitamin B9, and they are essential micronutrient for human health, because human and animals, they cannot synthesize them. They must obtain them from diet. So this is the structure of folate. So folate consists of three parts: tetrahydroterine, para-aminobenzoic acid, and the glutamate residue. So up to 6 glutamate residue is attached to this gamma link amino acid to make a polyglutamate tail. And this polyglutamate tail is very, very important for folate function, because folate dependent enzyme, folate functioning enzyme, they bind to the polyglutamate tail, and the folate transport enzyme, folate transporter, they bind to the monoglutamate tail. Therefore, this folate glutamate tail affect like cofactor activity as well as the transport activity, and indirectly influence the folate stability. So this pterine ring can exist in both like dihydrofolate and tetrahydrofolate form. Only the tetrahydrofolate form has the cofactor activity. So the N5 and N10 position of tetrahydrofolate can be attached to different C1 derivatives. They are at different different like oxidation stage. They can be attached to N5 and N10 enzymatically, and they are like interconvertible to one another form by using different enzymes. And this folate, they are very sensitive to photooxidation and oxidative damage at this 9 and 10 position. They can break down very easily to release the pterines and the PABA glutamate. So tetrahydrofolate and their C1 derivatives, they play a very important role in one carbon metabolism, where they donate one carbon to different reactions, as you can see here. Tetrahydrofolate interconverted to 5,10 methylene tetrahydrofolate by like it play very important role in photorespiration, and different folate derivatives, they are involved in different metabolic process. They are involved in the synthesis of like nucleotide purine and thymidine synthesis, vitamin B5, proteins, and amino acid metabolism, especially methionine and histidine. And they're also involved in the methionine amino acid synthesis, which in turn synthesize S-adenosylmethionine, which is very important source of like ethylene and polyamine. And the methyl group from this S-adenosylmethionine is a donor of different like protein, DNA, lignin, and choline synthesis. So coming to the folate biosynthesis, folate is present throughout the plant cell in mitochondria, plastid, vacuole, and cytoplasm, but they are only synthesized in the mitochondria. So GTP cycle hydrolase and the amino deoxyribonucleic synthase, they are the rate limiting enzyme for folate biosynthesis, and they have been used to like using metabolic engineering to increase the folate content in different crops. So and this is called like cytoplasmic branch is called pterine branch, and this amino deoxyribonucleic synthase, it is called power branch. So after synthesis in the pterine branch in this cytoplasm and PABA in chloroplast, they are transported to the mitochondria for tetrahydrofolate synthesis. Now, now polyglutamate tail is synthesized by the action of polyglutamate synthase, one residue at a time, to form a polyglutamate tail. This polyglutamate tail can be shortened or reduced by the action of gamma glutamate hydrolase, which is present in the vacuole. And in case of Arabidopsis, there are three polyglutamate synthase, but in case of tomato, there are only two, which one is present in mitochondria and another is present in chloroplast. And there are three gamma glutamate hydrolase in tomato, GGH1, 2, and 3. So in few of my later slides, I will be talking about this pterine pathway intermediate and PABA pathway intermediate. So like this is hydroxymethyl dihydropterine and dihydro monopterine and dihydropterine. So in few of my slides later, I will be talking about this. So just keep them like remember. So the functional importance of this polyglutamate tail has been done using this knockout mutant in Arabidopsis, where all the three FPGS gene exhibit like knockout of all these three FPGS result in defective proliferation rate, dark fish, and significant reduction in methionine level. So there are several strategies have been used to develop to improve the folate intake. So one of them is like production and consumption of folate-rich food, which require educational effort and change in the dietary habit. And second is folic acid supplementation and fortification. This technique has been very successful in developed country, but in case of developing countries, it is not like efficient because of lack of industrial food system and because of the recurrent cost. So the third one is folate biofortification in staple crop, though metabolic engineering. It has been very successful, and like it is a sustainable substitute, and it has been implemented in tomato and rice. In tomato, using this two rate limiting enzyme of folate biosynthesis pathway, GCH1 and ADCS, they were able to increase folate content almost 25-fold. And in case of rice, they use similar strategy and they have increased folate content almost like 100-fold. But because of the transgenic nature, they are like not in the market, and like because of consumer resistance and amid their health concern. So the alternate is to explore the genetic diversity, natural variation in the. It is a cost-effective and promising strategy to alleviate the micronutrient deficiency. So in our study, we have used this exploring the natural variation. So based on this background, we have asked few key questions. So one of them is to optimize a high throughput method for folate analysis, especially in tomato. And the second question, to identify tomato germplasm which is should be rich in folate. In our we have a large, huge collection of tomato accession. We had a huge collection of tomato accession, and the second objective was to identify tomato germplasm, which is rich in folate, and third objective was to understand the regulation of folate in this germplasm line. So moving to my first objective. So folate extraction involve different steps. So folate in food is very sensitive to light, temperature, heat, and they are present in very, very low amount. So folate extraction is very difficult. Folate extraction analysis is very difficult. It is a very challenging also. So tri-enzyme is the most commonly used method for folate extraction. So alpha amylase, protease, and conjugase, they're the most commonly used enzyme. So alpha amylase and conjugase, they break down the carbohydrate and protein substance to release the bound folate, and conjugase, it break the polyglutamate tail to monoglutamate tail for folate analysis. So in this is this method is like we have adopted this method from De Brewer, which is they have used for rice, which has very high amount of starch. So we have modified this method for our analysis. So we have changed few like amount of incubation, pH, and other things to make it suitable for our extraction in tomato. So another important thing is that folate purification. It is like very sensitive. So there are three different methods available for folate purification. One is solid phase extraction using Q-saffrage. Another is affinity chromatography, and third one is ultrafiltration using cutoff membrane filter. So the drawback of affinity chromatography is that it has very low affinity for 5-methyl tetrahydrofolate, which is like third most abundant folate form. So in our study, we have used SPE using Q-saffrage, but the problem with Q-saffrage is that it has a like 50%, 50 to 60% recovery rate, and we need to have very high amount of salt to elute the folate. And again, we have to dry that elutant, and if we are using LCMS, then they can create a problem in LCMS. So for our further study, we have used only the ultrafiltration. With ultrafiltration, it's very easy to use. Just we need like centrifuge, and we got almost like 95% recovery using 10 kilodalton membrane filter. So using this method, we analyze folate content in tomato leaf, tomato fruit, and different vegetables. So one of the interesting things, we observed that tomato leaf almost like more than 40-fold higher folate in compared to fruit, and the folate level measure in other vegetables were similar to the report in the literature. So using this method, we found 4 different folate forms. 5-methyl tetrahydrofolate was the most abundant folate form, and tetrahydrofolate was the least abundant folate form. So in summary, using this method, we found a better recovery, which is very sensitive, and extraction was fast compared to other method. And since we have used LCMS, so it was very accurate up to 5 decimal. And in single day, we can extract almost 40 sample a day. And this method is equally applicable to other vegetables also. So moving to our second objective, we have a large collection of tomato from different resources, like Bijou Setal is a private company, and Indian Institute of Vegetable Research, and National Bureau of Plant Genome Research, and TGRC UC Davis. So we have around 391 accessions. So we have grown them you can see in the picture. I'm not there. I just take the picture. And we have transferred them to field. And in this, you can see the fruit phenotype of the few accessions. So one of the biggest problem we have faced, loss because of wild boar, peacock, and squirrel. We have lost almost 60% of our crop. So using this method, this is just a representative picture. Using this method, we have analyzed folate content in 125 tomato accessions. So we have seen a wide variation in folate content. So from this, we have selected 2 cultivar. One is Arka Vikas, which is low in folate, and PKM, periculum mutant, which is high in folate. So and these avian pecan, they're like well-known cultivar in southern India, and they are grown in similar agroclimatic conditions. So we have re-examined the folate content in the next generation to check whether like the folate is again like high. So we have collected fruit from three different stage: mature green, breaker, and red ripe. So in case of tomato, these stage are very, very important, especially the breaker stage. When fruit start to ripe, they start to synthesize, accumulating carotenoids, and there is a burst of ethylene and respiration rate, and they start to accumulate like flavor also. So we re-examined the folate content in PChem again, although the folate content was lower compared to the previous season, but it's still it is more than twofold higher. So uh, in spite of having this environmental uh, seasonal difference, we have selected AV and PChem to understand the folate variation. So in the fourth subsequent experiment, we have grown this AV and PKM in greenhouse to avoid any environmental differences. So the on-vine fruit ripening of AV and PKM were almost similar. They reach the maximum size at around 30 days, and the mature green stage is around 32 days. From mature green to breaker stage, PKM reach around 7 days and AV reach around 4 days. So there was a difference of 3 days in AV and PKM, as you can see here. And fruit ripening is also accompanies with increase in sugar content. So we have measured the Brix content. So at the red ripe stage, PKM has significantly higher Brix compared to AV. So another a major difference we have seen is the decrease in ethylene content in PKM at breaker and red ripe stage. Like ABA, like ethylene AB was also almost half in PChem at the all the three different stages. And we have seen salicylic acid and jasmonic acid lower in like stage specific manner. So we did the like proteomic study, and we found some protein related to ethylene biosynthesis, which were like ACO1 and ACO6, which are like expressed during fruit specific ethylene synthesis. So they were very, very low in PKM1. And so we thought that reduce level of these hormones may be related to ethylene, because in one of the paper recently, the underproducer of ethylene also have low level of ABA, salicylic acid, and jasmonic acid. So although you can see the greenhouse grown plant have lower folate compared to the field grown, but still the folate content was very, very high, significantly high in PKM on all the three different stages. And the folate content was decreasing, steady decline from mature green to red ripe stage. The decline was faster in case of PKM, in case of AV. So to check whether the folate content is high only in fruit or it is like affecting the leaf also. So we have measured the folate content in leaf also. So leaf also saw the high level of significantly high folate content. And we have seen the 4 different folate forms. So the next question was whether high folate in PKM is because of precursor, which is PABA and terrenes. So like folate, PABA content was also high in PKM in all the 3 different stages. We could not detect the terrene derivatives because we presume that they are present in very, very low amount. But we could able to see hydroxymethyl terrene, which is like oxidized form of hydroxymethyl dihydroterrene. And low level of this is considered, okay, we'll go to one more slide. So low level of this is considered that salvage pathway is more efficient in recycling the hydroxymethyl dihydroterrene. As you can see here, the folate synthesis as well as the folate breakdown and salvage pathway is usually go parallel. So tetrahydrofolate, it is degraded to terrene and PABA glucose. So terrene in different enzymic state, like enzymatic and non-enzymatic state, is converted to dihydropterin 6 aldehyde. This PTAR enzyme is very efficient. It has very high activity. It can convert this to 6-hydroxydimethylpterine, and it can be used in the salvage pathway. But sometimes these enzymes fail to intercept dihydropterin 6 aldehyde. So they again oxidize to pterin 6 aldehyde, which are like this PTR has very like broad spectrum specificity. So if they fail to intercept this, it is converted to this by like light or non-enzymatic means. And again, 26 aldehyde is converted to 6-hydroxymethylpterine, which cannot be used for like salvage pathway. So low level of this means that salvage pathway is much more efficient, and it can be used for folate synthesis. So the next question was whether high folate in PKM is because of the expression of folate biosynthesis gene. So, but like we have not find much significant difference in our analysis. All the gene have more or less similar expression in case of AV and PKM. So we thought that maybe some like regulation is somewhere else other than this folate biosynthesis gene transcription regulation. So next we have measured the transcript level of gamma glutamyl hydrolase gene. So we found that GGH1, GGH2, and GGH3, their level was significantly low in PKM especially at the red ripe stage. After that, we have checked the GGH enzyme activity at GGH enzyme activity. So we found that GGH enzyme activity was very high at mature grain stage, and they started decline as the ripening progressed. So, but we don't find any significant difference in AV and PKM at the mature grain stage. But at the breaker stage, the GGH activity was significantly high in PKM, and they were significantly low in RR stage. And the folate polyglutamate level it is hypothesized that folate glutamate is the stable form, and they are less sensitive to degradation or catabolism. So we measure the folate monoglutamate and polyglutamate level in AV and PKM at the red ripe stage, and we found that PKM has significantly high polyglutamate level at RR stage, red ripe stage. So recently, not recently, one paper came where they have overexpressed this GGH enzyme, and they found that overexpression of GGH in vacuole reduced folate polyglutamate tail length, and the folate content was reduced almost 40%. So next, we have measured whether high folate in PKM may have any influence on carotenoid biosynthesis pathway. So we measure the carotenoid content in AV and PKM. At the red ripe, these pie diagrams show the proportional ratio. So at red ripe stage, AV PKM1 has almost 1.5-fold higher folate compared to AV, and they have lycopene and beta carotene almost 1.5-fold higher. So next, we examine whether high folate level, high carotenoid level in PKM is because of the transcript level of the genes. So at mature green and breaker stage, most of the transcript did not differ significantly. Their level was almost similar in AV and PKM. But at red ripe stage, few of the genes which synthesize lycopene were significantly low in case of PKM. So then we did the proteomic study, and we found some of the protein which help in carotenoid sequestration were high in case of PKM, like PAP3, keratin binding protein, and CHRC. They are known to play a role in carotenoid sequestration. So carotenoid in plastid, they are stored in lipoprotein-like structure, called the globules. So these proteins are component of those globules, and as you can see here, in tomato, they have overexpressed fibrillin protein. So overexpression of this result in increased carotenoid content and the carotenoid divolatize. Similarly, in tomato high pigment mutant, which accumulate high level of carotenoid, they also have high level of CHRC protein. So as like folate play important role in C1 metabolism, so we examine whether is there any influence on overall proteome. So we did use the GCMS to identify different metabolite. So we using GCMS we identify 69 metabolite which include amino acids, sugars, organic acid, and fatty acids, sugar derivatives. So as you can see in this figure, most of the metabolites were significant, and PCA analysis show the clustering of different metabolite at different stages of ripening and PKM. So they form separate cluster at each ripening stage. Then we have mapped these metabolite on metabolic pathway. So one of the interesting things we observed that most of the amino acid were significantly higher in PKM, as you can see the flat scale, and sugars and others, they are like vary in stage specific fashion. So like I have shown previously that in PKM the Brix content was very high. So in our proteomic study we found the sucrose synthase. So high level of Brix and sugar and sucrose in PKM1 may be related to high level of sucrose synthase. So we found two proteins related to glycolysis. So phosphoglycokinase and phosphopyruvate hydrolase. So they are the precursor for synthesis of like phosphoglycokinase. It is precursor for synthesis of serine, glycine, and cysteine. So we thought that high level of this may be responsible for the high amount of these amino acid. And the phosphopyruvate kinase may be responsible for leucine, valine, and alanine amino acid. And like we also found high level of aconitase. So we believe that high level of aconitase may be pushing precursor for synthesis of glutamate. And we found two more protein like glutamate synthase and glutamine synthase, which may be responsible for high level of glutamine and paraglutamate glutamate. And we also found a very low level of gamma transaminase. So it has been reported that over silencing of gamma transaminase increase GABA level in tomato fruit. So in our case, the low level of gamma transaminase may be responsible for high level of GABA. So in summary, a sample and high throughput method was developed, which is easy and high throughput, and which is equally applicable to other crops also. So tomato cultivar show almost threefold varies in folate level at the red ripe stage. And lower GGH activity and high polyglutamine level in RR fruit likely contribute to high folate in PKM1. Carotenoid sequestration protein seems to be key regulator of increased carotenoid level in PKM1. And PKM1 fruit have elevated level of three nutraceutical, folate, carotenoid, and GABA. So recently, Japan has launched this genome-edited tomato, which are very rich in GABA. So we also got a similar kind of result. So now here in the PEC lab, I am working on like phenolics content. So in the I have optimized two methods for analyzing phenolics monomer and the polymer, which is tannin. So I just want to give a brief introduction about phenolics. So phenolics are secondary class of secondary metabolites, which are involved in organoleptic criteria such as color, which is provided by flavanthriols like catechin, epicatechin, and hydroxychlorogenic acid, and bitterness and astringency is provided by flavanthriol, catechin, epicatechin, and tannins. And they also provide the colloidal stability to the cider. So they are the precursor of cider aromas and flavor like hydroxycinnamic acid, which provide characteristic flavor to Kingston, high price Kingston apple. And they are broadly divided into 5 different categories. Flavanthiol, which include catechin, epicatechin, hydroxycinnamate, which include chlorogenic acid and dihydrochalcone, chloridine, flavin like quercetin, and anthocyanins. So these are the like structure of few of the phenolics. So one of our previous lab member, he did screening of a large accession of Meles germplasm. Okay, I will go. So the cider industry is one of the fastest growing market in US, and there are not enough cider varieties which can be potentially used for hard cider making. The essential the requirement for high cider is like they should have a high amount of tannin. So he did with this objective, he screened this 158 Meles accession from USDA PGRU. And so he found that total phenolics content using Folate Chico. The total phenolics content varies from 0.39 mg per liter to 4.86 in juice sample. And these two, Northern Spy and Elizabeth, they are the commercial cider varieties which are used for cider making. But like interesting things, he observed that few of the varieties also have the high level of total phenolics, which can be used for potentially for cider industry. So he did the like total phenolics analysis, and I optimized method for to analyze the phenolics monomer using HPLC. So using HPLC, we identify around 19 phenolics monomers in all these 14 varieties cultivar. And if you see the individual phenolics, many of them has higher phenolics monomer compared to like catechin, like all the flavanthirols compared to the commercially cider varieties like Alice Bitter and Northern Spy. And we did the PCA biplot analysis, and we found this procyanidin B1 and chlorogenic acid are the factor which determine the PC1 and PC2 variation. So the other objective was to optimize the method for HPLC. So just a brief introduction about procyanidin and or condensed tannin. So they provide mouthfeel to the cider, and if their mean degree of polymerization is 6 to 8 or more, they provide astringency, and if they are lower than that, they provide bitterness. So they are the oligomer and polymer of catechin and epicatechin. Epicatechin is present in both, like as you can see, terminal unit as well as the extension unit, and the catechin is present only on extension unit. So we use the fluoroglycerol method, which hydrolyze a tannin polymer to monomer, and it release the terminal unit as a free form, and it form adduct with the extension unit. So using this method, we have measured the tannin content in porter perfection. So in lifeless tissue, so we have found that tannin content in porter perfection is 15 mg per gram dry weight, and the average molecular weight is 3000 Dalton, and mean degree of polymerization is around 11 mer. I'm almost done. But this is acclamation. I would like to thank my Professor R.P. Sarman, Dr. Y.C. Lakshmi, and all the lab member and the funding body Department of Biotechnology, which provide fund for this for this project, and Council of Scientific and Industrial Research for providing fellowship to me during my doctorate. And thank you all.
Any questions?
Kamal, has there been a effort in India to focus on growing more tomato varieties with higher folate? With that, now that you've screened all those accessions and you know which ones have higher folate naturally, has there been any sort of effort to commercialize those more?
Actually, this like PKM, it is already a commercial variety, and it is grown in South India. But this paper came recently, so maybe they will read this paper and hopefully they will like implement this.
Because I understand, in a lot of places, genetically engineered crops are, you have a lot of
Actually, in
cultural and political consumer issues, baggage. Yes, baggage would be a good word. And so I know in India that's been true with like the eggplant. And so I'm wondering about just kind of might be more interest in some of the naturally high cultivars versus the genetically engineered ones.
actually people don't know about this like PKM, but now maybe they will start growing this more and more.
Thank you very much. Kamal.
Thank you so much.
This has been a production of Cornell University, on the web at cornell.edu.